Good afternoon to you all, and thank you very much, BIPSA, for this opportunity. Oh, it's a shame I can't see the faces of the people I'm talking to, but I'm sure most of you know me. I work at the Noguchi Memorial Institute for Medical Research. Yeah, so I've been away because of COVID. I'm working virtually, and I'm currently in the US, but still actively working. So I'm not a staff of Pine Bio, but I found the group online as I explored what to do with my sequence data, apart from just understanding variants in the sequence. I wanted to explore a bit more about sequence data. So I went online and found this group, and I decided to enroll in one of their courses. I think it was data science, and I think it was just a 10-session program. But I was so excited about the way they were able to quickly transform somebody like me with very little computational skills into analyzing data. And so I decided to stay on and take on a project. So basically, I decided to work on COVID because it is the main issue on board. So together with another colleague, we worked on how certain drugs that have been repurposed for COVID treatment work. We know that COVID-19 has plagued the world, and there's very little information or drugs available. I think currently there's only one drug that is available, remdesivir. And there are other drugs that are being repurposed. One of such drugs is Zolutinib, which is known to inhibit the JAK-STAR pathway, which is a signaling pathway that is involved in immune responses and also replication of the virus, differentiation of the virus, and the death of the host cells. So any drug that will inhibit this pathway is also going to block the downstream activities of the pathway. And Zolutinib is known to inhibit the JAK-STAR pathway. So our project was to use Zolutinib as a standard drug whose pathway is known, or its mechanism of action is known, and how it will act as an anti-inhibitory drug for, as an anti-inflammatory drug for COVID treatment. So ours was to take another drug, and this drug was a calcium blocker, amlodipine, to see how such a calcium blocker may act to also prevent severe COVID diseases. So what we did was to take cell lines treated with the virus and cell lines treated with the virus and these two different drugs. And we had data from gene expression from these treated cell lines. And the analysis we did was to look at differential expression between these different groups. So cell lines treated with just the virus, and then cell lines treated with amlodipine, and cell lines treated with Zolutinib, to see differential gene expression between these three groups. And also to look at pathways that were enriched as a result of differential expression between these groups. So what we found in the nutshell is that amlodipine, which is a calcium blocker, is also able to inhibit the JAK-STAR pathway, just like Zolutinib does. So there was genes that were differentially expressed, and these genes inhibited or enriched certain pathways. And for Zolutinib or amlodipine treated cell lines, there were downregulation of these genes in the enriched pathways. So basically, SARS or the virus is enriching these pathways by upregulating certain genes, and these drugs are inhibiting the pathways by downregulating certain genes. So for a calcium blocker like amlodipine, we're able to determine how it may be acting to reduce the effects of COVID infection. And of course, there were other pathways that were either upregulated or downregulated, but this was the main finding of our study. And what excited me the most is the way I was able to quickly look at the expression table and then do analysis such as dimension reduction using PCA or principal component analysis, looking at normalization of my data quickly, and then passing it through analysis or algorithms like the DSEQ or DSEQ for expression level or differential gene expression, and further doing the gene ontology to look at pathways that were enriched for somebody with very little knowledge of programming or coding. And so I think that the group is here. They will tell you more about how these pathways work or how their platform works. So let me hand you over to Ilya, the CEO of Pine Biotech, to do the rest of the